This thematic talk will be continued by Dr. Jayasree. So Dr. Jayasree obtained a PhD from University of Kerala, Trivandrum in 2002. She completed a postdoctoral fellowship from Monash University, Melbourne, Australia. She is currently working as a scientist G in Sri Chitra Tirunal Institute for Medical Sciences and Technology, Trivandrum. Her research focuses on optical neural modulation and optogenetics, MR imaging, nanobiophotonix.
That's okay. So I have only 10 minutes. So she will take she will take 2 minutes and she will ask me to stop. So while he is loading, I can tell you 1 minute the story. So previous speaker, Dr. Lakshmi Bala was talking like you should dream and you should take your career when you feel like that. So just for as a comment, I am telling I was in this field. I don't know how Asima left me anyway. I started my career in 1987, but came to this optical field in 1996. But and I have 3 children, 3 boys. I brought up all of them, but in between I took a career break. Otherwise, it would have been very tough for me. Then I rejoined only at the age of 45, and I don't have any hesitation to tell you that I am now 60. So if I am in the state service next year, I will retire because ours is at 65. I have maybe 5 more years. So I am from Sri Chitra Tirunal Institute. So because I am using pen drive, he has cut off me from this slides change. Anyway, so Asima has introduced to you the topic. I will just show you some of my work directly going into that. I just introduced my institute. Many of you, I assume that you don't know what is Sri Chitra Tirunal Institute. It is the only institute, only hospital under the Department of Science and Technology, Government of India, and all other hospitals or healthcare institutes are under the Ministry of Health. So I will just quickly give some of our work. The first one I have included is the ultra-sensitive sensor platform for circulating tumor cells. The circulating tumor cells means when the cancer is metastatic in nature. If it is the spreading type, metastatic means if it spreads from one place to other place, then the tumor cells sometimes shed into the blood circulation, and it will be taken away from the primary center to another center. So if you can find out whether these cells are available in the bloodstream, then the doctors can find out whether the cancer has metastasized or has started spreading. So this is good in the prognosis stage, not for the diagnosis stage, because when the patient is in the treatment stage, whether it is treating. So this introduction and all I am skipping. I haven't removed the slide because I wanted to introduce these terms to you, but I don't have time to stick to the slide. So just keep watching and you can read, okay, later on. So this is what liquid biopsy that means. Originally it is we take the biopsy and now it is like we can have the liquid. So the body fluids taken from that and we can use it for as a biopsy sample. So this is the prototype that we have developed. This is just like a centrifuge tube. You can just see this is the 50 ml centrifuge tube, and what is the crux is the filter where we have used nanobiophotonix, and it has been modified with the biomarkers using nanographene and the golden nanoparticles just to enhance the Raman signal, and it has been conjugated with the antimarker for the circulating tumor cells so that it will be stuck on the filter when you centrifuge it. This is the nano tag, how you do it. So we have used 2 different types of nano tag. One we will mix with the blood, so and another one is coated on the filter. So both ways it makes a sandwich so that none of the thing is left. So that is one of the thing where, and all the parameters have been optimized, and this is initially after a centrifugation, all the red blood cells will be taken away and you will get the filter, and you can take out the filter because as I shown you this This has three different chamber. You can remove the top chamber, and this center chamber you can remove and take out the filter, and you can place that filter on the microscope, and you can take it either with the immunostaining, with the immunofluorescence staining, or with the Raman. Here I have done some Raman experiments, that is surface enhanced Raman, because we have graphene and gold. The signal enhancement is there because of the presence of graphene. The D and G bands, the two bands we could identify, because the other markers will always be the same for biological many of the molecules. Only a few biological, like amide 1, amide 2, amide 3, etc. So that will be identical. So here the intensity of the D and G band have been optimized, and you can just see up to 5 cells we could get the sensitivity of up to 5 cells. So generally it is maximum up to 5 cells available per ml. So that much sensitivity we could get. So this is one of the technique that this is using fluorescence technique. You can just see after the staining you got the cells captured. So this I am just skipping, and another technique I am quickly going. All the questions you can ask me during the coffee time, okay? And this is another one where we have developed a Alzheimer's disease marker. That again here it is nanobiophotomics, and the introduction on this you can go and read. And this is how we make the substrate. So here again we coat the substrate either glass sheet or the tin coated glass sheet with the gold nanoparticle, again with the Alzheimer's marker. So when you talk about Alzheimer's marker, there are established or identified markers, that is A beta 1 to 42 and tau protein and total tau proteins. So these proteins have antibodies and anti-markers. So that we will use as for selectivity. Otherwise, every proteins will get bind to this. So here again you can just see we have recorded the Raman signal, and you can see up to the concentration of femtomolar level we could get the Raman signal, but the attomolar level we could not get impressive signal. So from this we can quantify from the signal intensity the number of the concentration of the biomarker. So this is for the A beta 1 to 42, and the next slide is for P tau 181 protein. So these are two major proteins or the biomarkers identified for Alzheimer's disease. So you just note that these markers are not commonly found in the blood. So we are taking the blood here that I will show you, but usually it is present in the cerebrospinal fluid because Alzheimer's disease is a disease that affecting the brain. So the body fluid connected with that is cerebrospinal fluid. But there will be marker in the blood, but that is very, very, very less. So we have to have techniques which can capture that small concentration of biomarker. So that is very important. So these are the clinical results of some 15 or 30, I don't remember. And this is the A beta. These ratios are very important. A beta 42 by 40, A beta, P tau 81 by A beta 42, and total tau by A beta 42. So these are the differentiation or classification. Here you can see the first one is Alzheimer's disease. Second one is mild cognitive impaired patient, and this is the control. So there is a clear classification, even though there is a overlap between these groups, but you can get the clear classification. And with this we are also planning, we have already developed, but the database we are yet to build because we need clear cut separation between all the three. So this is also possible. You can bring out point of care diagnosis devices. And extending that work, this is not in the liquid biopsy, but we have also used this nanobiophotomics for treating the Alzheimer's disease. You can just see these are the fibrils. For example, those who know what is this A beta it forms as fibrils in the neurons and it makes plaque. That is why it impairs the functions of the neuron. So we got this neuronal fibrils unfolded using this type of nanoclusters. I am not going into the material part, only I am talking about the optic side. And another major challenge is it should cross the blood-brain barrier when it is used as a treatment technique. It should go and reach the brain. So there is a big challenge whenever you are giving anything to the brain. The brain is highly protected. It has the blood-brain barrier surrounded. So the materials is materials are modified to cross the blood-brain barrier. All those techniques I am not talking here. You can go and see my publication or any publication. So here also you can just see this is in the cellular, this is the control cells. This is along with fibrils and the material. So the fibrils have been completely unfolded here. So we have developed Alzheimer's disease animal models and we have tested this. You can just see the behavioral study. The red one is the material from the material is here. It is gold cluster, the Alzheimer's disease plus material plus L-DOPA. Here we have used L-DOPA as the brain targeting one. Otherwise, it won't go to the brain. Here you would have seen that these animals, the signal is not from the brain. Only this one has the signal from the brain. That is because this material has L-DOPA with that. So that will target the material to the brain, whereas this in these two animals it goes somewhere else because it does not have brain targeting. So this one of these have been published in this, and you can just see this is a biosensor which we have published in 2013. This is a urea biosensor. This is here again we are using nanobiophotomics. So here the sensor has been made in such a way that the sensor fluoresces in the infrared NIR region, so that any biological fluid has interference in the visible region, it will autofluoresce. So that you should avoid. For that we have tuned the emission of this material in the NIR region, so that when you take the blood as the sample, its fluorescence will not interfere. So you can use the whole blood. You don't want to separate it into plasma and serum. So this is a material and for urea detection, where the fluorescence from the fluorescence from the sample quenches. So it quenches with respect to concentration that can be quantified. Here you can see the clinical comparison, and the red one is with our technique. Here again the fluorescence technique is being used. This is the selectivity of the one, and at the same time this is the same concept. We are using same technique for the sensing of multiple analytes at the same time. So that also you can do. Here we have introduced WOPI and creatinine. We are using two different peaks of these things. One is increasing, one is decreasing. How much time I have? Done? You should have given me a warning. Okay, so this has been developed into a mobile app-based sensor, and I'll take 2 more minutes. So these are some of the old work where we have used fluorescence spectroscopy, which Asima was talking now. So some of these things you can just see the fluorescence spectra from the oral cavity, from the brain samples, etc. have been collected, and it has been classified based on the excitation wavelength that you have given. According to the excitation wavelength, it will have corresponding emission. That is, if you are exciting at 360 degree, and you will get correspondingly what all parameters will fluoresce. That is all established. So quantifying those things, you can differentiate the abnormal and normal tissue that we have used for oral cavity diseases, brain tumors, etc., which I have just put it here. I'm skipping, and I think you would have had the flavor of what is the work, and now the rest you can read. Let me see anything additional. I think all of this the same thing. it is not well organized because I prepared the, this is the same. Okay, then this is the last one, sorry. Here we have taken the coronary bypass, during coronary bypass surgery, we take the artery and vein from your radial artery or the saphenous vein from the thigh or the internal mammary artery. So we have just checked whether these, we are taking the graft from the same patient, because if the patient has any other comorbidity with these vessels, will have comorbidity, will have some additional parameters which have to be looked into, which usually is not. So we have studied the infrared imaging, and we found that some of these grafts have high lipid profile, which we need to be looked into. That is one of our observations. So with that, I stop here. Thanks.